Welcome to our module on nerve tissue. So the fourth type of tissue in the body is the nerve tissue. The other three types of tissues include the epithelial tissue, the connective tissues, and the muscle tissue. All three we've talked about in a separate module. The nerve tissue is the main structural component of the nervous system. And when we talk about a nerve tissue, we talk about a collection of cells that give rise to the nerve tissue. There are two major forms of cells that tend to combine together to form the nerve tissue. This includes the neurons and what we call nuclear cells or neural supporting cells. So let's start by talking about the neurons. Neurons are the functional units of the nervous system. Between the neuron and the nuclear cell, in terms of the actual function of the nervous system, which is in the function of communication between different aspects of the entire body, the neuron is involved in doing that particular function. And the reason why this is so is because the neuron, just like a muscle cell, is an excitable cell. So excitable cells are cells that are able to generate action potential and as a result help in signal transmission. So neurons are excitable cells that generate action potential and they are specialized in that signal transmission that we see across the entire nervous system within the body. Talking about the structure of a neuron, the neuron as a cell has three major parts to it. It has a cell body, or what we call the soma. It has a dendrite or a bunch of dendrites attached to it at one pole. And then at the second pole, we have the axon. Usually a single axon tends to emerge from it. This is a picture of the entire component of the neuron. This is the cell body at the center. And just at the upper pole of the cell body, you could see different projections branching out of it. These projections are known as dendrites. And then finally, at the lower pole of the soma or of the cell body of the neuron, we see this singular, long or elongated projection or process. This is the axon. Here you can see how it tends to run all the way down. And each of these different components have different functions. We'll talk about that in the next session. So starting off with the cell body or the soma, the cell body of the soma is the central portion of the entire neuron. It is the part that contains the nucleus. Remember that the neuron is a cell, and just like every other cell, it has its own form of nucleus that is involved in production of DNA and transcription and translation of mRNA substances into protein. Aside the nucleus found centrally in the cell body, we also have organelles that are sparsely distributed around the nucleus. Some of these organelles will include the mitochondrion, as shown in the image. You can see the pictures of mitochondrion around the nucleus. And then we have some special form of endoplasmic reticulum, what we call the nasal body. So nasal body is the endoplasmic reticulum of the nucleus. And then we have unique cytoskeleton and pigments found within the neurons. So the pigments that we see within neurons are usually lipofuscin. Then we have specialized cytoskeleton found within the neurons known as neurofilaments. So the function of the cell body is to act as a focus for the cellular metabolism. So all the metabolic processes that would take place within the neuron usually takes place within the cell body, and that's the major function. The second component of the neuron is the dendrites. The dendrites are multiple processes or projections that tend to emerge from the cell body at a particular pole. The major function of a dendrite is in its ability to receive action potential from neighboring neurons. Remember that the sole purpose of a neuron is in the function of action potential generation and transmission. So once action potential has been generated from a particular neuron, that action potential tends to be sent to another neuron for what we call signal transmission. And the part of the neighboring neuron that receives the action potential that has been produced is the dendrites. The dendrites practically are multiple projections of the neuron cell body that is present there to receive action potential. The last component of the neuron is the axon. The axon is a single projection of the neural cell body, and it is in opposite poles to the dendrites. The axons tend to be involved in conducting action potential. So the same way in which we have the dendrites receiving action potential that has been generated by a neighboring neuron, the axon of that particular neuron would send the action potential to the neighboring neuron. So axons are involved in sending action potential. Dendrites are involved in receiving action potential. There's some unique structures of the axons that you should know about. First of all, is the fact that the connection point between the axon and the cell body is known as the axon hillock. The axon hillock is this enlarged portion of the cell body that tapers down to form the axon. What is important to note about the axon hillock is the fact that it is the site of initiation of action potential. It's the point where, what we call the trigger zone for initiation of action potential. The axons, once they have been formed, tend to project all the way down towards a neighboring neuron. And another unique property of the axon is the fact that it is wrapped around by special lipid structures known as myelin sheets. Myelin sheets are lipid structures that wrap around the entire length of the axon. So you can see the myelin sheet shown in the image here. And what you notice about the myelin sheet is that they don't form a continuous layer around the axons. They tend to be discontinuous. And that discontinuity we see of the myelin sheet is made possible by it eventually gives rise to what we call nodes. So there are different nodes between different myelin sheets along the entire axon, and those nodes are known as nodes of Ramberg. So you can see this is one node here, this is another node. So these are all nodes, internodes or nodes of Ramberg between each of the myelin sheets. So this is important because the myelin sheet, first of all, insulates the entire axon. Secondly, the presence of those nodes between each of the myelin sheets allows for faster transmission of action potential across the entire axon. So how is this possible? So once we have a node present between different action myelin sheets wrapping around an axon, once action potential is generated at the axon hillock, this action potential tends to jump from one node of Ramberg to the other. So you can see it's jumped from one point to another point, jump from this node to this node, from this node to this node, and that way we have faster transmission instead of the action potential just running through the entire length. That jumping motion of the action potential across the nodes makes conduction of action potential faster. The third property of the axon is the fact that it lacks a nasal substance. So nasal substances or nodes are found within the cell body of the neuron. Nasal substances are the endoplasmic reticulum of the entire neuron, and it is absent in the axon but present in the cell body. And then the last property of the axon is in a presence of special cytoskeleton found within it. So these cytoskeletons are a combination of neurofilaments and microtubules. So we have microtubules found in the axon. The microtubules present within the axon is present there for the function of transmission of protein molecules, whether we're talking about neurotransmitters or otherwise. Protein molecules found within the entire neuron can be transferred from the cell body to the axon, to the end of the axon, what we call the axon terminal, or from the axon terminal back to the cell body. And we have microtubules involved in doing that. So the particular microtubule that transmits protein from the cell body to the axon terminal is known as kinesins, and the type of transport is known as an anterograde transportation from the cell body to the axon terminal. Then the microtubules involved in transmitting proteins from the axon terminal back to the cell body is known as dynein microtubules. Dynein is involved in what we call the retrograde transport from the axon terminal back to the cell body. So these are the various properties of the axons, and you can see how it is unique when we compare that to the rest of the components of the neurons. How do we classify neurons? We can classify neurons in many or multiple ways. We can classify based on structures. We can classify based on the function of the neuron. We can classify based on the target of the neuron. We can classify based on the neurotransmitters that the neurons tend to produce. We can classify a neuron based on shape, and we can classify based on axonal properties. In terms of this particular module, we'll just focus on two major forms of classification, which is the structural classification and the functional classification. So structurally, we can classify neurons either as a unipolar neuron, a pseudo-unipolar neuron, a bipolar neuron, or a multipolar neuron. And then functionally, we can classify neurons into either a sensory neuron, a motor neuron, or an interneuron. Let's start with the classification based on structure. We have the unipolar, bipolar, pseudo-unipolar, and multipolar neurons. So this usually talks about the different forms of processes that emerges from the cell body. Remember that typically a typical neuron has two major forms of processes. The dendrites usually tend to be multiple dendrites on one pole, so on one pole of the cell body, and then the axon being another process on the second pole of the um of the cell body. So based on the amount of processes coming out from a particular pole of a cell of the cell body of a neuron, we can classify a neuron either as the unipolar, bipolar, pseudo-unipolar, or multipolar. So unipolar, as the name implies, is just a single process coming from one pole. You can see just the cell body staying there and then just one single process, really an axon coming out of it. Usually we don't see this in adult structures. We tend to see unipolar neurons in embryonic structures, but they are typically seen in embryonic structures and even in non-human or in organs or organelles or organisms that are non-vertebral in classification. Then we have the bipolar neurons. Bipolar neurons, as the name implies, are neurons that have two poles emerging from the cell body. So we have one pole facing up, the other facing down. All right. One pole is involved in receiving, the other one is involved in giving or conducting. And we tend to see bipolar neurons in special organs like the eyes, like the nose, that's the olfactory center of the nose, like the gustatory center of the tongue, and then the auditory center of the ears. They all have bipolar neurons involved in special sensation. Then we have the pseudo-unipolar neurons. Pseudo-unipolar neurons are unique in the sense that they are neurons that appear to have just a single process, but this single process that emerges from the cell body would eventually branch to form two processes. So that's why we call them pseudo-unipolar. They appear to be just unipolar in nature, but that singular pole or singular process coming out from the pole of the cell body will branch off into two. So it branches off into two, one going centrally, the other going peripherally. So one receiving, the other giving. That's practically what those poles tend to be. We tend to see pseudo unipolar neurons, particularly in sensory as sensory neurons involved in somatic sensation. So areas where we see somatic sensations being perceived, we have pseudo unipolar neurons playing a role in it. And then finally, we have the multipolar neurons. Multipolar neurons depict a typical neuron, and that's because most of the neurons present within the nervous system are multipolar in nature. A multipolar neuron has multiple processes emerging from different poles. So we have dendrites being multiple, and then a single axon being the singular process occurring at the extreme pole. So this is the typical depiction of a multipolar of a neuron. That's the multipolar neuron in terms of how it looks like structurally. Right, let's talk about the classification based on function. So this is simply broken down into sensory or afferent neurons. So neurons that tend to receive impulses, particularly impulses relating to sensations. Then we have the motor neurons or efferent neurons. These are neurons that are involved in sending impulses. So they usually send impulses particularly to muscle structures and to glandular structures. And then we have the interneurons. Interneurons are just neurons that are lying between the motor and sensory neurons. They're there to confine and regulate neuronal activities present within the central nervous system. The second half of this particular module will focus on the second aspect of the nerve tissue, would be neuroglia cells. So neuroglia cells are non-excitable cells. So in contrast to neurons, they don't undergo excitation. They don't produce action potential. The major function in the nervous system is for support. So they tend to support, they tend to insulate, they tend to nourish, and they tend to protect the neurons. The neurons are like they are like the key players in the nervous system. Just think about neurons as the 10 man or the 11 man in a football match, and then the neuroglia cells as the fans, the supporting team or supporting teammates in a football team. So we have five major neuroglia cells to know. Each of them have various functions. We have the astrocytes, which are the most abundant. We have the oligodendrocytes. We have the ependymal cells, the Schwann cells, the microglia cells, and sometimes we talk about the stellate glia cells as well. But most of these cells are just five in number: astrocytes, oligodendrocytes, microglia, ependymal cells, Schwann cells. Each of them are distributed across different aspects of the nervous system. Some are found exclusively in the CNS, some are found exclusively in the PNS, some are found in both of them, and they have separate functions. Let's start with the astrocyte. Astrocytes are the most abundant nuclear cells, and they have a number of different functions we'll talk about. They are star-shaped in appearance, and they possess long radiating processes. As you can see, the reason why they are star-shaped is because of this elongating processes that tend to stem out of them. They are found in the entire part of the central nervous system. So they're found in the entire central nervous system, and their major functions would include, first of all, physical support to the neuron. They help in supporting the neurons physically. They also help in protecting the neurons by acting as a blood-brain barrier. They produce the or form the blood-brain barrier that helps in protecting the neurons. And whenever the neuron is being damaged, the astrocytes are involved in the repair of a neuron, or usually a process we call gliosis. And then in nourishment of the neuron, the astrocytes tend to serve as stores for glycogen, and they also help in removing excess neurotransmitters. The second important nuclear cells are the ependymal cells. Ependymal cells are simple columnar cells in nature. They are located in the entire central nervous system, and they are localized to a particular part of the nervous system. So the central nervous system comprises of the brain and the spinal cord. And each of these structures are not entirely solid structures. They are hollow structures that have spaces between them. So within the brain, there's a space there called the ventricle, as shown in the image here. This is the entire brain, and you can see this tiny space in the center of the brain. This tiny space is known as the ventricle. And then within the spinal cord, there's also a space in the entire center of the spinal cord known as the central canal. Each of those spaces are lined by ependymal cells. So the ependymal cells tend to line the ventricles of the brain, and they also tend to line the central canal of the spinal cord. And that's their major function: the lining of the lumen or the spaces within the central nervous system. The second function of the ependymal cell is in the production of CSF fluid. These spaces that we talked about, the ventricles of the brain and the central canal of the spinal cord, are not empty spaces. They are filled with fluid. The particular fluid that is found within the spaces and is known as the cerebrospinal fluid. And the cerebrospinal fluid tends to be produced or processed by the ependymal cells. Ependymal cells tend to have cilia and microvilli found in their apical surface, and this cilia and microvilli is present there for the movement and flow of cerebrospinal fluid across the lumen of the central nervous system. The third nuclear cell is microglia cell. Microglia cell is just another fancy way of saying CNS macrophage. So just the same way we have different macrophages in different organs or tissues of the body with different names, the microglia cells would be the CNS macrophage found in the nervous system. So macrophages, for example, in the liver would be known as Kupffer cells. Macrophages in the lungs is known as alveolar macrophages. Macrophages in the kidney is known as mesangial cells. So the one in the CNS is known as microglia cells. So, and they are the smallest nuclear cells in the entire body. They are also located in the entire central nervous system. They are involved in performing a number of functions that are peculiar or pertaining to immunity. So some of the functions that the microglia cells do would be phagocytosis of cellular debris. So whenever there's a damage to cells and the repair is going on, microglia cells will be involved in phagocytosing the cellular debris. Whenever there's an infection within the entire brain, like meningitis or encephalitis, microglia cells will be there to mop up those microorganisms causing the infection. And then finally, microglia cells tend to promote inflammatory reactions going on within the CNS. The fourth nuclear cell is the oligodendrocytes. Oligodendrocytes are cells that are limited to just the white matter of the central nervous system. They are found in the central nervous system, that is in the brain and the spinal cord, and they are found in the white matter portion of the CNS. So what does white matter mean? So white matter portion just refers to the aspect of the central nervous system that contains axons that are myelinated. So myelinated axons tend to give rise to the white matter of the central nervous system. So those areas where there are myelinated axons is where we see oligodendrocyte. And the reason is simple. The function of the oligodendrocyte is in its ability to produce myelin sheath. Is the part of the nuclear cells that produces the myelin sheath that wraps around the axon. So it makes a lot of sense that it is only going to be found in a place where there's axon and where these axons are myelinated, because it is the structure or the cell involved in the production of the myelin sheath. So oligodendrocytes tend to have a long, numerous processes that tend to help in myelination of multiple axons at once. So you can see this is a single oligodendrocyte, but you can see the processes helping in myelinating at least more than one axon. You can see one axon is another axon is another a third axon. Each of these axons are myelinated by a single oligodendrocyte. The last nuclear cell we'll talk about in this module will be the Schwann cells. Schwann cells are located only in the peripheral nervous system. They are located outside of the central nervous system. So nerves like the musculocutaneous nerve that we'll talk about in the upper limb or the peroneal nerve talk about in the lower limb, the examples of PNS nerves, or cranial nerves like the olfactory nerve or optic nerve are examples of PNS, which are the peripheral nerves. These nerves also tend to have myelin sheets wrapped around them, and the particular structure that, or the particular cells helping in the formation of myelin sheets for peripheral nerves tends to be Schwann cells. So Schwann cells are the counterpart, or Schwann cells are the counterpart to the oligodendrocytes in the CNS. So oligodendrocytes are involved in myelination of nerves in the CNS. Schwann cells are involved in myelination of nerves in the PNS. And they are found in the PNS also for the purpose of repair. So the same in which astrocytes helps in repairing structures of the central nervous system whenever there's the damage. Schwann cells are also involved in repairing or neurons in the peripheral nervous system whenever there's damage. So let's talk about the structure of a nerve tissue. So we'll do something similar to what we did when talking about the structure of a muscle tissue. So nerve tissues are usually bundled together and wrapped around by a number of connective tissue. And we'll talk about it starting from the smallest structure going up to the largest structure. So starting with axons. So we start with a collection of axons. So an axon itself is known as a nerve fiber. An axon, each of the axons tend to be wrapped around by a connective tissue we know as the endoneuron. So endoneuron wraps around a single axon or a single nerve fiber. So when we have a collection of nerve fibers, together, which is a collection of an axon and its individual endoneuron together would be wrapped around by a second connective tissue, and that connective tissue is known as the perineum. So when we have a perineum wrapping around the collection of nerve fibers, we call that the nerve fascicle. So a nerve fascicle is collection of axons or collection of nerve fibers wrapped around by the perineum. And then finally, collection of nerve fascicles wrapped around by the last or third layer of connective tissue is known as, which is called the epineurium, would give us the nerve tissue. So nerve tissue is the nerve fascicles wrapped around by an epineurium. So just to repeat what I said before, a single axon will give rise to a nerve fiber, and that will be surrounded by an endoneuron. When we have a collection of nerve fibers with its endoneuron, we call that a nerve fascicle, and that is surrounded by the perineum. And then finally, when we have a collection of perineum, that will give rise to a nerve tissue, and that is surrounded by an epineurium. So let's conclude this module by talking about two major important clinical significance to the topic of nerve tissue. The first would be multiple sclerosis. So multiple sclerosis involves the problem with myelination in the CNS. And the second would be Guillain-Barré syndrome. Guillain-Barré syndrome involves problem with myelination in the peripheral nervous system or the PNS. So in multiple sclerosis, we have a problem with myelination, usually problems with oligodendrocytes being destroyed, and as a result, we have reduction in myelin sheath formation. And as a result, this affects the conduction process of action potential across different axons in the CNS. And this will lead to symptoms like fatigue, depression, vertigo, vision problems, sexual dysfunction, bowel and bladder dysfunction, muscle-related symptoms, which is the commonest, and then cognitive dysfunction also emerging. So in this particular image, we have a problem with a nerve fiber in terms of myelination. This is the typical depiction of the pathology we know as Guillain-Barré syndrome. In Guillain-Barré syndrome, there's a problem with the Schwann cells, and Schwann cells are involved in myelination of the PNS nerve fibers. So if there's problems with that myelination occurring or damage to the myelin sheets formed, we could have poor conduction of action potential across the peripheral nervous system. Across the peripheral nervous system, that is across peripheral nerves, and that will lead to problems with muscle movement. Particularly, people would come down what we call ascending paralysis. So the person could wake up at night or wake up all of a sudden and just find that they can't move their legs or move their hand as a result of that problem, demyelination or problem with myelination that we see in Guillain-Barré syndrome. And that concludes our module on nerve tissue.